Students, welcome to your channel The Anatomy Canvas. In today's class we are going to discuss with the histology of the eye. So first of all, the eye, it is about 24 millimeter in diameter, located within hollow bony orbit of the skull, and histologically it is formed of the three tunics. So the outermost is the fibrous tunics, which consist of the cornea anteriorly and the thick fibrous coat, the sclera posteriorly. Then the middle coat, which is shown in blue color over here, and this coat consists of the iris, then the ciliary body, and then the choroid, collectively called as the uvea or the uveal tract. And then you can see this yellow color, this is the innermost coat, which is the retina. So the three main coats histologically of eye is formed of three main coats, and it is present in bony orbit. It is about 24 millimeter in diameter. So this is the diagram of the eye, just to have an idea. Outermost layer anteriorly, this is the cornea. Then here you can see this is the iris, and the chamber which is present between the cornea and the iris is called the anterior chamber. The window which is present between the iris is called the pupil. Behind the iris you can see the lens is present, and in between the lens and iris you can find the posterior chamber. Here in this area you can see the ciliary body, which continues as this is the area of the ora serrata, and the ciliary body continues as the choroid over here. Innermost coat is the retina, which you can see on inner aspect, and in this area you can see the fovea centralis is present, and this is the area where optic nerve is present. This chamber is the vitreous chamber. So the eye has the three chamber: the anterior chamber, which is present between cornea and iris, the posterior chamber, which is present between iris and the lens, and the vitreous chamber, which is present between the outer layer of the lens to the retina. So this whole chamber is covered by the vitreous body. Then first of all we will discuss the cornea. The cornea, a word mean horny. It is anterior 1/6 of the fibrous coat. It is avascular, very important MCQ. It is avascular, it is transparent, and it consists of 5 layers histologically. These 5 layers are the corneal epithelium, the Bowman's capsule, which is also called anterior limiting membrane, the substantia propria, Descemet's membrane, also called posterior limiting membrane, and corneal endothelium. So we will do all of these one by one. First, you can see in this diagram that this is the outermost layer. This is the corneal epithelium. Then beneath the epithelium in this area, this is the higher magnification taken from this area. So here you can see the epithelium, then the Bowman's membrane. So this all, which is also called the stroma, this is the substantia propria, the third layer. Then at this area, from this area, this section is taken at higher magnification to show you the presence of the Descemet membrane, and then the corneal endothelium. So 5 layer of the cornea: the epicorneal epithelium, then the Bowman's membrane, the stroma or substantia propria, then the Descemet's membrane, and endothelium. So corneal epithelium is basically, this is stratified squamous non-keratinized epithelium, consists of 5 to 6 layers of the cell. It is very sensitive. It is rich in the nerve ending. It has the capability to rapidly regenerate after any abrasion, and this regeneration usually takes place within a week. And the process by which this regeneration takes place is by the gliding movement of the adjacent cells to fill in the gap. Later on, mitotic active cell will be present to cover these aberrations. So this is the outer layers, and outer side the squamous cells are present, which are attached to each other by the zonula occludentis, and apical microvilli are also present. Another important point is that nuclear DNA is protected from the ultraviolet rays and X-rays by the nuclear ferritin. This is again another MCQ. Next to the corneal endothelium is the Bowman's membrane. This is relatively thick 10 millimeter basement membrane, and it consists of the ground substance and irregular fibers of the reticular fibers. So you can say this is the basement membrane of the epithelium, the reticular lamina, and along with it small amount of the ground substance. Collectively these are forming the Bowman's membrane. This membrane is strongly PAS positive, shows that the it is rich in the polysaccharides. This membrane is actually not a membrane, but it is the outer layer of the substantia propria of the cornea. So here you can see this is the substantia propria or stroma of the cornea, and this is the outer layer of the substantia propria. So next, very important, is the substantia propria, that is forming nearly 90% thickness of the cornea. This consists of the parallel and regularly arranged bundles of the type 1 collagen fibers, and this is because of this arrangement which gives transparency to the cornea. It is because of this arrangement of the collagen fibers that they are regularly arranged and they are parallel to each other, which gives transparency to this area. Then is the descemet membrane. Here you can see this one is the descemet membrane over here. So this is nearly 5 micrometer thick. It's a thin membrane. It covers the substantia propria posteriorly. So this is the substantia propria. Anteriorly present, anteriorly was the Bowman's membrane, and posteriorly is the descemet's membrane. Then is the corneal endothelium. This is a single layer of the squamous epithelial cell. This is not actually endothelium. The word is a misnomer because it has no relationship with the blood vessels, and outer to this anterior chamber is present, in which anterior chamber the fluid is present, and this fluid passes by the help of the microvilli to the corneal endothelium, and which will give the nutritional supports to the cornea. So these are the 5 layers of the cornea. The first of all, the corneal epithelium, then is the basement membrane, that is the Bowman's membrane, then is the substantia propria, also called the stroma, then is the descemet's membrane, and corneal endothelium. Another important point is an electron micrograph of the corneal stroma. This electron micrograph shows parts of 3 lamellae and portion of the corneal fibroblast. So this is the corneal fibroblast which is present. This is one of the lamellae. This is another lamellae, and this one is another lamellae. So the collagen fibers in adjacent lamellae are at right angle to one another. In one lamellae, all the collagen fibers are parallel to each other, while they are at right angle to the collagen fibers in the next lamellae. So here in this area you can see I mark with a marker to show you. This is the one lamellae. From here to this area, this is the another lamellae, and from here to this area, this is the third lamellae. So fibers over here, they are transversely cut, and these fibers and these both are longitudinally cut. So all the fibers over here are parallel to each other, but at right angle to the fibers of the adjacent lamellae, and this arrangement is important for transparency of the cornea. Next is the sclera, which is outer coat posteriorly, and it covers posterior by 5 by 6 of the fibrous coat. It is collagenous, consists of type 1 fiber, and fibroblasts are also abundant in it. The extraocular muscles are attached to this area. So this its thickness is 1 millimeter at the posterior pole, 0.3 to 0.4 millimeter at equator, and 0.6 millimeter towards the edge of the cornea. Between the sclera and the choroid is the large layer of the loose connective tissue with the elastic fibers and numerous melanocytes and fibroblasts. Histologically, this sclera consists of the 3 layers. The outermost, which is close to the extraocular muscle and the periorbital area, this is the episclera, which is loose connective tissue to which the extraocular muscles are attached. Then the middle coat, which is also called the sclera proper or the Tennant's capsule, consists of the type 1 collagen fibers. These are the thick fibers which are present over here, and then is the suprachoroidal lamina. This area, this is the suprachoroidal lamina. It is also called the lamina fusca. It contains, in addition to collagen fibers, this also cleared elastic fibers as well as the pigment cells which are present over here. So this area is adjacent to the choroid over here. So you can appreciate the cross section, the longitudinal section of the skeletal muscles are extraocular muscles. Then you can see the sclera, the outer part which is episclera, the middle part which is the sclera proper, and the inner part the suprachoroidal lamina, also called the lamina fusca. Then this area is the choroid. So middle vascular cord, also called the uvea, it consists of the anteriorly is the iris which is present over here. Then it continues as the ciliary body, and this ciliary body continues as the choroid. So this is the vascular cord of the eye. This is the middle cord of the eye, and it is also called the uvea or the uveal tract. So the choroid, the middle vascular cord, the uvea consists of the choroid. Here you can see this area, this is the extraocular muscle. Then you can see the sclera proper over here, and this area is the choroid, and then you can see the retina. So these are the three muscle cord. This choroid again consists of the four layers. The epichoroidal or suprachoroidal layer, which is in contact with the sclera. Then the middle layer, the vascular layer, the capillary layer. Some books mention these two layers, the vascular and capillary layer, as the choriocapillary layer as well. And then is the Brooks membrane, which is attached to the retina. So choroid or epichoroidal or suprachoroidal layer, here you can see this one outer layer is the loose pigmented tissue composed of fine transparent sheet of fibroblast, rich network of elastic fibers, and large flat macrophages. Then is the vascular layer, which contains medium sized vessel. So this black color is because of the pigmented cell which are present, and these empty spaces over here shows you the larger blood vessels which are present over here. Then this black color is because of pigmented cells which are melanocytes. Then you can see in this diagram also that third layer is the choriocapillary layer, which consists of the fenestrated capillaries, and they supported by delicate connective tissue containing melanocytes, and these capillaries are source of nutrients and oxygen to the retina. So you can see next to the choriocapillary layer is Brooks membrane, and then is the basal layer of the retina. So this choroid by this layer is attached to the retina, and it is the source of nutrients and oxygen to the retina. So the Brooks membrane, then it is a glassy membrane, you can see, also called lamina vitrea as well. It is 1 to 4 micrometer thick, composed of network of elastic fibers located in the central region, and it in turn consists of four layers. This is also very important. First of all, from choroid side, the basal lamina of the capillaries. So as I have told you that in the choriocapillary layers, fenestrated capillaries are present. So the basement with the basal lamina of the endothelium of this layer forming the first layer of the Brooks membrane. Then is the layer of the fine collagen fibers, then second layer of the elastic fibers, then third layer of another layer of the collagen fiber, and then basal layer lamina of the pigment epithelium of the retina. So these five layers collectively they are forming the Brooks membrane or lamina vitrea, and through this membrane the nutrient passes from the capillary layer to the basal layer of the pigment epithelium of the retina. So this is all for the today's class, and we will do the next topic in the next class. The references for today's class are Laikus and Siddiq, the Histology Atlas and text by the John Quera, and Histology text and Atlas by the H. Ross. Thank you. Thank you very much for your attention, and Allah Hafiz till the next class.